As already mentioned, the primary principles of phototransduction also apply to a second type of specialized photoreceptors found in the human retina, the cones. While less sensitive to light than rods, cones are responsible for color vision and eye color sensitivity and function best in relatively bright light, such as during a normal sunny day. Cones are also able to perceive finer detail and more rapid changes in images than rods. In the human eye, cones are generally one of three types, each with a different visual pigment, able to sense different wavelengths of the light spectrum. Shortwave cones, or S cones, detect blue light with a peak wavelength of around 420 to 440 nanometers. Middle wave cones, or M cones, detect green light with a peak wavelength at around 535 to 550 nanometers. Long wave cones, or L cones, detect red light with a peak wavelength of around 565 to 580 nanometers. For comparison, rod cells have a peak sensitivity at around 498 nanometers, roughly halfway between the peak sensitivity of the short and middle wave cones. This spectral sensitivity and difference in the signals received from the three cone types allows the brain to perceive a continuous range of colors. All cone receptor types contain GPCR protein called photopsin, also known as cone opsin, which is able to transduce light in a similar manner as rods through the conversion of 11 cis-retinal to all trans-retinal. The color sensitivity stems from the fact that the photopsin protein differs by a few amino acids for each cone type, thus allowing light absorption at different wavelengths. So how does actual color vision occur across a range of colors with only red, green, and blue sensors? The key factor in being able to represent a range of colors is the fact that these spectral sensitivity curves are spread out and overlap quite a bit. Thus, different wavelengths of light elicit different levels of responses from each of the different cone types. This is how you can see purple light even though you don't have a purple cone. As you can see, we can tell apart different pure wavelengths of light by looking at the relative activation of the three cone types. We call this trichromat, or three-color vision. Of course, most objects in real life don't reflect pure wavelengths of light. The light from the sun has a wide range of wavelengths, and the light coming off of different objects has a spectrum of its own. So the responses of the three cones, and the color we perceive, depends on the overlap of the spectrum of the light with the absorption of the cones across all three cone types. Of course, we can't represent a whole spectrum of light with just three cone outputs, so many different spectra of light will produce the same perception of color. For instance, these patterns of light both produce the same outputs from the S, M, and L cones, so we perceive them as the same even though the underlying pattern of light is very different. We call these different patterns that elicit the same percept metamers. The screen you're watching this on right now takes advantage of this effect. If we show you this flower on the screen, you can see it as yellow, and it looks a lot like a flower you would see in real life. However, in real life, a complex spectrum of many wavelengths of light reflect off of that flower and into your eye. The monitor you're looking at now can't hope to actually produce all of those wavelengths, so instead it relies on trichromat vision to do the next best thing. If you zoom in on the screen, you'll see tiny red, green, and blue spots, which are tailored to excite your three cone types. To produce the percept of the yellow flower, the monitor just needs to excite the S, M, and L cones by the same amount as the flower's real spectrum did. Much like our visual system, most everyday devices use the RGB color model to produce the perception of color. Interestingly, humans and other mammals have evolved trichromacy based on pigments partly inherited from earlier vertebrates. But in fish and birds, for example, 4 pigments are used for vision. Because these animals have an extra cone with a different absorption spectrum, they're able to distinguish colors that would look the same to us. For these tetrachromat animals, 2 spectra of light would have to produce the same responses in all 4 cones to produce the same percept. Interestingly, it turns out that there are some humans that are tetrachromats as well. Some women have 2 slightly different copies of one of the opsins on each X chromosome, giving them the ability to resolve more colors than most people. Conversely, some people have fewer than 3 cone pigments. For example, certain mutations in the photopsin GPCR proteins can result in vision with only 1 or 2 cone types. This leaves the individual without the ability to perceive certain colors, which is commonly referred to as color blindness. In the case of red-green color blindness, or deuteranopia, individuals lack M-type cones and thus can't distinguish between red and green because these colors are metameric, perceived the same way, because they excite the remaining cones identically. It should be noted, however, that since rods are more light sensitive than cones, colorblind individuals generally have normal light sensitivity in dim light. During night vision, also known as scotopic vision, rods are far more active than cones. The light's too dim to activate the cones. This is why it's hard to perceive color at night. In the bright outdoors or an artificially illuminated store or office, however, cones are active and rods are bleached or inactive. Thus, color vision is dominant. This is also known as photopic vision.